Hello everyone, I am Raphael Mourad, uh, Associate Professor at the University of Toulouse, and today I will make a short lecture on large language models for genomics. So here is first a quick introduction to large language models. I will first talk about deep learning as a branch of AI. So basically you can consider that actually the AI as artificial intelligence was mostly developed during the 50s, 60s, 70s. And then during the 80s, 90s, 20,000, there was machine learning that was introduced where you can train a model based on data, and this model can take decisions. That's why you call it machine learning. And then from the 2010 to now, there has been a very important development of specific machine learning algorithms that are called deep learning algorithms that corresponds to very deep neural networks. So there has been a very important success of deep learning since 2012. For instance, if we take the example of computer vision with ImageNet challenge classification, before 2012, the classification error when you want to predict whether an image is a cat, a dog, a house, or a car, for instance, and you want to build a classifier basically predicting, given an image, predict what you have in the image. The error rate was quite high, 28% or 26% to an 11. And for instance, when you compare that to what an eye can do, you could say at this time that an eye does not make much error compared to a classifier. But with deep learning in 2012, starting in 2012, there has been a very important drop in the classification error. So there has been new algorithms basically based on deep learning that were where the classification error was getting lower and lower and lower until we obtain something that can be even better than an eye, a human eye. Uh, so so basically, there is when you make algorithms and you train them, when you train models, I mean, when you train models based on data, so you give examples of data, and then the model will be trained on that. Traditionally, you have machine learning models where you provide data as an input, for instance, the image of a car, and there is an expert who will provide the features. Basically, if you have an image, you need features to describe, which are basically variables that describe the object in the image. So basically, what is a car? A car basically is composed of wheels, it's composed of windows, it's composed of an engine. And if you can consider features, all of that as features, and then you can count the number of wheels you have, for instance, then if you have zero wheels, then zero wheel, then you don't have a car. So you can make a feature, and that will be these features will be very informative in predicting whether you have a car or not. And then based on that, you can use traditional machine learning, traditional machine learning models like regressions, random forests, super vector machines. It can also be a neural network like the perceptron, and then you can predict whether you have a car or not. With deep learning, there have been a very important progress with deep learning when it comes to, for instance, analyzing images, because the idea behind deep learning is you don't provide features that will be extracting from the image. You will the model will build by itself the different features. So basically, the model by itself will recognize that wheels, windows, the engine, for instance, are key features of a car, and it will predict. So basically, the model will identify what makes a car a car. I mean, what are the different components of a car, and it will identify it. So you will not need knowledge from an expert, because usually you need expert knowledge to identify features. So by deep learning, we mean that we use neural networks, and neural networks here are deep. They correspond to a stacking of different layers. So here you have as an input an image of a digit 4, and then you have 4 layers when you have linear transformations, often followed by nonlinear transformations. And then at the end, you will predict with the classifier whether you have 0, 1, 2, 3, or 4. Okay? And so the most, I would say, the simplest key component of a neural network is, so here you have a simple one-layer neural network where it takes different features that are different variables, x1, x2, x3, etc., xi, and then you apply weights on the different variables. So basically, it's a linear transformation of the input variables, that are the xi, by weights yij. So you will transform, you will use the linear transformation to modify your features exactly as it is done with a linear regression. And and then you have an activation function that can be linear, meaning that you have no change in the output, or you can use sigmoid, or you can use tan function, so you can make it nonlinear. So this is just a one-layer neural network, okay? But then you can stack different layers. So for instance, here you only have one neuron. Here you have multiple input features, and you just have one neuron. Here you can have multiple neurons. So here it's one layer containing many neurons, okay? And for each neuron, basically, you apply the same formula, which is a linear transformation here, and then a nonlinear transformation with the function f. So it's often a linear transformation and then a nonlinear transformation. And then this is for one neuron, and you do it for many neurons, okay? And all the neurons corresponds to different transformations of the input features. And you can stack. You can consider, okay, I've made my first layer of neurons, and then I can make another layer of neurons that will transform again the first layer of neurons, etc. And then you have an output layer when you have classification here. So here, for instance, if I take the example of an image, so you have an image of kids, and then you want to predict labels. For instance, do you have a kid, do you have a cat, do you have a house or whatever? Then you'll stack many transformations here, many layers. Okay, so this is basically what it does. It is a stacking of different layers. So there are, of course, so what we saw here is we call that a multi-layer perceptron, where you have different layers of dense neurons. We call that a dense layer because you can see that there are many, there are all possible connections between a neuron here and the input features here. But then you can also have other transformations such as you can have other neurons such as convolution, rectification, normalization, etc. Softmax, which are different transformation of the data. And so what's very important with, so we will not go into convolutional transformations or recurring neural networks or LSTM or other kinds of neural networks. We'll focus more on the attention mechanism so that will introduce a transformer model. So in the attention mechanism, it consists in the following. For instance, imagine that you want to translate this phrase in English, how was your day? And you want to translate in French, which is comment se passe ta journée. So when you do that, you can see that when you want to translate to comment, you can see that how is a key word, of course, how translate to comment. But actually in French, you can translate how to different French words. And the choice of comment will depend not only on how, but it will depend on was. So basically, the translation from how to comment, it depends on was. Okay, so basically how it's translated to comment, but it depends on was, because there are different, you can also translate to quoi or quel. So there are different French words. So basically it means that when you translate to comment, you have to pay attention also to another word, which is was here. That's why you call that attention. That is the attention mechanism. So same thing when you want to translate. So also something interesting is in English you have four words. The corresponding sentence in French it comprises five words. So basically, ce is introduced here, and it relates to both how and what. So that's why actually you need both words and the attention is high for how and what. And the rest of the sentence is not important to know that you have to say so. Also pass. Pass is was, but also how. But you can see how, here that your to ta actually is the translation is almost direct. You could also translate to ton, by the way. So you can see that mostly you will choose ta. That's why mostly the most important, the highest weight, attention weight is given to your, okay? And journée corresponds mostly to day. So you have basically, when you translate, for instance, with attention mechanism, it means that you don't translate one word to another word directly. You will pay attention to the context, okay? That's the idea behind it. And then you can think about self-attention, which is if you want, for instance, to predict a word in the sentence giving other word in the sentence. So this is the same idea. So for instance, imagine that you want to say le chat dort sur le tabouret, which means the cat is sleeping in the chair. When you talk about the cat, so it's an example in French. When you talk about the cat, in French, a cat is considered as masculine, as a boy, for instance. So when you translate, you don't say it, you use masculine, and then why masculine? Because the cat is considered as masculine. So that's why you translate directly to that. So you have to, when you want to predict the next word, you have to pay attention to which word before. That's why it is self-attention. So it will tell you where are in the sentence the most important information when you want to predict the next word. And how do you compute that? Basically, you have basically, so you'll take an input feature like a sentence here. See that girl run. So you have a sentence. It will be, so here an input data X. And you will transform this input data as usual with a neural layer here. So it's just a regular neuron, but you have to compute the attention weights. The attention weights will be a transformation also of these X input variables. And here you will combine, you will compute weights between each words in the sentence, basically. So that's the point here. You will compute basically for girl, you will compute all of the weights of the different words in the sentence. So you compute the attention weights, and then you will weight each word in the sentence using this attention mechanism. And then that's why it's very important, because the attention mechanism is very used in the transformer architecture, and the transformer architecture is at the basis of the modern language models, large language models. So there are two kinds of large language models: causal language modeling. I mean, large language models that are trained for causal language modeling, and large language models that are trained for masked language modeling. So basically here, for instance, you have the sentence all the very, and there is a mask, so it means that you want to predict the word behind the mask, and the right word is best. So causal language modeling is, given these three words on the left, you will predict the word best. Okay, so it's unidirectional and you predict from left to right. And you also have the masked language modeling, where any word in the sentence can be masked. So it's random. They're all masked randomly. And basically you will, so for instance, here, if there is a mask here, it means that you will take into account the context on the left, the context on the right. So it's bidirectional information. A very famous mass language model is a BERT model. So inside it, this is a stacking of many transformer blocks. And I'd advise you, because I mean, I don't have time here in this lecture to explain deeply what is a transformer block, a transformer architecture, but I strongly advise you to read more about that. So the input data, for instance, you have this sentence. So CLS is the beginning of the sentence. How are you, I guess, doing today, end of the sentence? Okay, how are you doing? And then you will mask one word. For instance, it can be you or it can be are, and then we will try to predict it. And the model will predict it. It will be you, they, your. Okay, most of the time it should give highest probability for you. That's what we'll predict. There is also the so I wrote causal, but it's masked, sorry, it's masked language modeling. And sorry for that. Sorry, it's causal language modeling. What did I say? So here it's masked language, here it's causal language modeling, so there is no mistake. And here we want to predict, given a sentence, what is the next word. Okay? For instance, the GPT-1, the first GPT, which was the GPT-1 model was developed and was already giving years ago good result. It was improved into GPT-2 and then GPT-3, and that we know more nowadays, and GPT-4 that gave rise to ChatGPT. So here you can see that the number of parameters increased strongly. GPT-1 was composed of 100 million parameters with 12 decoded layers, 768 hidden layers, and a context size of 512 tokens. And then, given that it was already not working badly, it was already giving good results, there was improvement to researchers worked a lot to improve it to GPT-2 with 1 billion parameters and GPT-3 with more than 100 million parameters. And then by increasing the number of parameters, improving the architecture of the model, and also increasing the size of the data that you give to the model, researchers obtain better and better models. So for instance, if you can take the example of GPT-3, it has been trained on the file, which is a very important data. Very important data. It has all the Wikipedia articles, many books, I mean, a lot of books, a lot of web pages to be trained. Also, for instance, it was trained on archive that contains a lot of scientific articles, YouTube subtitles, a lot of Gutenberg, it's for the books. So PubMed is from scientific articles. It was trained on a lot of text. And for instance, it gave rise to ChatGPT. Okay, so this is where most people heard about GPT, because results were really good. Let's go now to deep learning for genomics. So the human genome is basically composed of 22 autosomal chromosomes, 2 sex chromosomes, and mitochondria. It is roughly composed of 3.3 billion DNA letters, 80 that can be only 80 GC. And here, just below, I gave you an example of a DNA sequence. So the question is, when you have a DNA sequence, how do you? So when you have a human language, such as English, you have sentences with words. So each word can be a feature, so it's pretty easy to distinguish features. However, when it comes to DNA, you have letters, but you don't have words. So one way of making words from DNA can be to use k-mers. That has been used a lot before deep learning, also before large language models. And you can, so you, basically the idea is like if you have a sequence here, you can split it into the different k-mers. Here, 7 mers. And then you can, for instance, so it will be the new words of your DNA sequence. So basically, you can consider that your DNA is a paragraph made of words, and here the words will be k-mers. So for instance, based on this idea, you have DNA BERT, which uses the BERT architecture, but which was trained on DNA sequences that were taken from the human genome. So the model extract the different k-mers. So here k-mers of 3 letters. And so here it is here. So you have the DNA sequence, it's split into k-mers of size 3. And then you will mask also different k-mers of different words. This data will go inside the transformer architecture, the BERT architecture that is composed here of 12 transformer blocks. And then you will predict, basically your model will be pre-trained to predict the word inside the mask, to predict the missing k-mers. And then there has been improvement of DNABERT with DNABERT 2, for instance, trained on much more data. Also recently, there was the publication of nucleic transformer. That is a very big model that was trained not only from the human genome, but from thousands of human animal genomes. And you can see here the performance of the model to predict different types of data and answers. So basically, so here is when you fine-tune the model on classification, just to say. So when you pre-train the pre-trained model is not able to predict whether you have an answer or histone modification or whatever on your DNA sequence. It's just, the model is just trained to predict a word in a sentence that has been masked. But then you can fine-tune this model, for instance, on the classification task to predict whether you have an answer, whether you have a histone mark, or whether you have a promoter, for instance, okay? So I developed I would say, a toy model, the Mistral DNA model, which is a large language model based on the Mistral architecture, and that you'll be using this model during the different tutorials that I made on Google Colab. It is a causal language model, so basically, given a DNA sentence, it will predict the next word, meaning that the next k-mer. So your sentence is based on different k-mers, okay? And you'll predict the next k-mer on the right, for instance. It uses a byte per encoding tokenizer, so it's a way to build the different tokens of different words. And how does it work? And you can look at Wikipedia, it's very well detailed on Wikipedia. Imagine that you have a DNA sentence that is like that. And then you can see that you have a word that is repeated many times. So CT is a k-mer, basically of size 2, that is repeated 3 times. So the algorithm will see that CT is repeated 3 times, and it will, because so it is very frequent, and it will replace it with a new variable called Z. So this way it will identify, for instance, k-mer of size 2. And then if you want to say a k-mer of size 3, maybe you can see that ZT is found many times, and if it's the case, you have ZT, then you will make a k-mer of size 3. Visual DNA also includes comprises a sparse mixture of experts that reduces the number of parameters that are used during inference. So it's also a way to make the model more efficient during inference. So just to give you an example, how does it work? It takes an input sequence of, for instance, AT, GET, GET, A, CG, CGG. It will split it into different tokens, and it will predict the next token, that is TGGG. Okay, so this is the way the model is pre-trained. Okay, so let's look at the applications that we also during the Google Colabs. So first application, you can fine-tune basically your model for classification. How does it work? So your pre-trained large language model is not made for classification. You have to add a classification node on the top, and it can be easily done using the transformer library. So as you can see in the Google notebook, so basically you add the classification node. So you add new parameters to learn from the model. And then, of course, you need to provide label sequences, specifically DNA sequences that are labeled as one and DNA sequences that are labeled as zero. So for instance, you can consider that DNA sequences that labeled as one are DNA sequences that bind to the protein CTCF. And you can consider that a DNA sequence is zero if it does not bind to CTCF. Okay, and then you can make a classifier based on that by fine-tuning the model on label sequences. And for instance, after you can imagine, okay, I take a chromosome, I split it in small sequences, and then I want to predict for all of the sequences on my chromosome whether it binds or not to the sequences. So here is just a schema of CTCF ChIP-seq data, ChIP-seq experiment, where when you have a peak, it means that you have CTCF protein that binds here. And then this is what you predict. So for instance, here I just imagine that we predicted this peak, here we predicted this peak, but here there is a missing peak we didn't predict it because the model can fail sometimes. Another application of this kind of model is we can predict the impact of a SNP, for instance. So your large language model will take a DNA sequence and input, and it will compute the embedding of the DNA sequence, which is basically a vector of numbers. So basically, it's a way to represent a DNA sequence that are made of letters as a quantitative variable. And we can assume that if two DNA sequences are functionally very different, then they should have different embeddings. So for instance, we can imagine that here I have a DNA sequence here. The letter here is a C. And the same sequence with the same sequence, instead of a C, I have a T. So basically, it is what is called a single nucleotide polymorphism. It's a SNP. So here it changes from C to T. You can take this sequence, send it to the LLM and compute the embedding. Okay? You can do the same with this sequence and you compute the embedding. Okay? And then you can compute the L2, so the Euclidean distance between the two corresponding embeddings. And if the L2 distance between the two corresponding embeddings is very large, you can assume that the SNP has a strong impact on the function of the sequence. So that's what I tried here. So I took 50 or 100, I forgot exactly the number. I took SNPs that are in exons, so that can modify proteins, and SNPs that are in introns that are predicted not to modify protein sequence. So you expect that the SNPs in exons should be more functional, should have higher impact than SNPs that are in intron. And then when you compute the L2 distance here, you can see that indeed the Lange-Joven model was able to see that exon SNPs, or SNPs in exons, have a higher impact than SNP in introns. So you can see that it works actually. Let's look at now synthetic DNA sequence generation. So it works only for causal language models, just to say. So you want to predict even, for instance, so how do you do? Imagine you want to predict a synthetic DNA sequence. What you can do is you can just tell to the model that you have the beginning of the sequence, so meaning that no DNA letters, and then it will predict a token. And then if you take this token, you'll predict another token, etc. And then this way, you will generate an actual DNA sequence. If you do it from a large language model that was trained on the yeast genomes, then you'll predict DNA sequences that are similar to yeast genome sequences. So for instance, here, that's what I've done. I generated 5 DNA sequences from a model that was pre-trained on the yeast genome. And then what I wanted to check is, are these DNA sequences totally new? So I took the first sequence and I blasted it. I used BLAST. And BLAST showed no matching to any known DNA sequence. So you can see that it's indeed a new DNA sequence. And then what you can do, and what you can see as what you will see as the last Google Colab tutorial, is a DNA sequence optimization. So the idea, it consists in finding the sequence with the highest or the lowest score predicted by a pre-trained model. So imagine that you take an LLM that was fine-tuned. So you take misread DNA that was fine-tuned for promoter prediction. So the model saw label sequences. Some sequences are promoters, some sequences are not promoters. You give that to the model, you fine-tune it for classification, and then it means that now you can take any DNA sequence you want and predict whether it has a high score of being a promoter, if it has a low score of being a promoter. And then you can use that for optimization. So the idea of optimization is the, op the, op the optimization will take your neural network that is just a function for the optimization algorithm, and it will start from an initial sequence. You can take a random DNA sequence, and it will modify your DNA sequence little by little until it finds a DNA sequence that has a very low or very high score, depending on what you want to do. So here, just to say, it doesn't mean that we found the best of the best, meaning the global optimum, the best of the best DNA sequence, but just to simplify it, I just say this is the best sequence. But if your algorithm might not find the best sequence, it might find a very good sequence, meaning a local optimum. So for instance, what you can use is a black box optimization algorithm, such as evolutionary algorithms. Just keep in mind that this is a discrete optimization, so it's not a continuous optimization. You just have letters like A, T, G, C. So your space at a given position is you can just have A, T, G, or C. And then what you can do is you can give as an input an initial sequence, and then your model will, for instance, your algorithm will use crossover, so mix sequences with each other. I mean, when you have as an input just one sequence, it will not start like that. It will just add point mutations, like it will add just SNPs, so just point mutations. It will change a letter A into a T or a letter C into a G. It will do that in parallel for many DNA sequences. And then after it will use crossover. So it will combine. So when you mutate one, so you take a sequence, you mutate it, you take the same sequence and mutate it, but at a different position. And then at the end, you have a population of different sequences, okay? And then you will use crossover, okay? So it means that you will take a portion of one sequence and you'll combine with another sequence. So for instance, you have a sequence in blue, and sequence in white, you cut it here, and you will combine the blue part with the white part. And here's the white part with the blue part, okay? So it's a way to add randomization inside, randomness inside the process of generating DNA sequences, basically. And when you have generated DNA sequences, at each step, at each iteration of the algorithm, you will take your neural network to predict whether it's a promoter or not. And this way, and you reiterate. So this way you will mutate, you will modify your DNA sequence little by little until the score, its predicted score, for instance, of being a promoter will increase until it will reach an optimum. So I hope you like this video, and you will see the different Google Colab for the different tutorials. Bye bye.